Welcome back. Today we're tackling, well, one of science's biggest data challenges, really. How do you get a computer system to understand both our language, human language, and the very specific languages of science? Things like complex molecular formulas or maybe those long protein sequences.
Yeah, that's been a tough nut to crack. Historically, models were good at one or the other. They understood the narrative, the text, or they understood the structure, like a smile string for a molecule.
But not really both at the same time.
Not easily. The way a smile string is put together, its syntax, is just fundamentally different from how we structure sentences, the semantics. And scientific knowledge, it's locked up in all these different formats. Your general AI models, well, they often stumble on that very specific domain-level grammar.
So our deep dive today focuses on something called SciReasoner. It's a new scientific reasoning foundation model that's specifically built to try and bridge that exact gap.
And the scale is, well, it's quite something. It was pre-trained on a massive data set, 206 billion tokens.
206 billion.
And the key is that corpus successfully blends standard scientific text with those specialized scientific sequences we talked about.
And the result is a single system that can handle a really wide range of scientific tasks.
An unprecedented breadth, actually. The aim is to support workflows covering, I think the paper mentioned, up to 103 tasks across 5 major capability families. So the mission seems to be unifying fields that were previously pretty separate, computationally speaking.
Okay, that scale, 206 billion tokens, it suggests they didn't just, throw random data at it. There must have been a strategy behind constructing that data set. How did they actually align natural language with something like a FASTA sequence for a gene?
Right, that's the core innovation in the pre-training phase. They curated this unique multi-representation alignment corpus. It wasn't just scientific papers, it was natural language alongside pure scientific sequences, DNA, RNA, protein sequences, plus those molecular strings like SMILES, IUPAC names, it's a standardized chemical naming, and selfies, another representation, and even formats for material science.
So the model wasn't just reading about molecules or proteins, it was learning from their actual structural codes.
That's it. It's learning the blueprints directly alongside the descriptions. To give you a feel for the mix, protein sequences made up about 33%, chemistry data around 30%, DNA was 18%, RNA 16-ish%, and materials about 3%.
Okay.
That broad, but also very structured exposure across domains seems critical for the kind of generalization they're reporting.
But just having all that data, that massive pre-training, gives it knowledge, right? How did they then teach it to actually reason like a scientist, not just spit back patterns it memorized?
Good question. That comes after the pre-training. There were 3 main stages to shape its reasoning abilities. First, standard supervised fine-tuning, SFT. They used about 40 million instructions there to align it with specific research tasks.
Okay, so telling it what kind of answers are needed.
Exactly. Then came a really interesting stage, annealed cold start bootstrapping. This was specifically designed to encourage the model to produce long-form chain of thought, or COT explanations.
Chain of thought. So for anyone listening, that means instead of just giving the answer, say the result is X, the model has to show it's working. Lay out the steps it took to get to X.
That's the core idea. It forces the model to generate a logical, step-by-step rationale, makes the process more transparent and hopefully more accurate. That second stage is about getting it to show verifiable steps.
And the final stage.
The final stage was reinforcement learning, or RL. Here, they use task-specific rewards to really fine-tune the reasoning, aiming for deliberate, verifiable scientific thinking based on whether those steps were correct.
Managing reasoning across, 103 different tasks, that sounds like it could be really inefficient. If I just need a quick property prediction, I don't want the model spending ages generating a complex 10-step derivation.
Right, the efficiency versus thoroughness trade-off.
Okay.
They addressed that with something they call adaptive reasoning. The insight was, well, not all scientific questions need that deep step-by-step thinking process.
Yeah.
So they basically categorized tasks into 2 modes. Instant tasks, like simple property predictions you mentioned, use the original SFT data for direct, fast answers. Think of it like a quick lookup or reflex.
And the other mode?
The other mode is for thinking tasks. These are the complex ones, maybe designing a new molecule, figuring out a chemical synthesis backwards, retrosynthesis. For these, the training data is replaced with those COT-augmented versions that encourage detailed, elaborate reasoning.
So it allocates the heavy-duty reasoning only when needed.
That's the principle. It separates the tasks to use reasoning capacity efficiently, keeping things fast for simpler queries, but allowing depth for complex problems.
That makes sense. But how do you make sure that distinction, instant versus thinking, doesn't cause it to miss something? Like maybe a seemingly simple prediction actually needs a little bit of reasoning.
Well, the distinction is based on task complexity and whether verifiable steps are typically needed for that kind of problem. And the robustness gets reinforced later, especially during that reinforcement learning phase you asked about. That RL stage needed some clever reward structures too.
Let's talk about that RL stage then. Giving a model a score for its output sounds tricky when the output could be, I don't know, a complex molecule, a gene sequence, or just a number for a material property. It's not like grading an essay, right?
Definitely not. It's not a simple right or wrong in many cases. So they developed task-grouped rewarding. They basically clustered the tasks based on the type of scientific output and used different reward strategies.
Okay, like what?
Three main types. First, distance-based rewards. These are for predictions where the answer is a number, a quantitative value. How close is the prediction to the known value?
Makes sense for property prediction.
Second, matching-based rewards. These are for tasks like scientific retrieval or pulling specific information out of text. How well does the extracted information match the ground truth? And third, this one's quite neat. Scientific tooling verified rewards. Here, the model's output is actually checked for validity using external professional scientific software packages.
Ah, so using established scientific tools as the judge. That grounds the model's output in real-world scientific practice.
It really does. And there's another challenge with RL they had to solve. Often, scientific validation gives a very hard binary result. It's either right or wrong, true or false. That kind of sharp signal can make training these big neural networks quite unstable.
So what do?
They implemented something called scientific reward softening.
Softening the reward. How does that work?
Instead of just giving a 1 for correct or a 0 for incorrect, they transformed the task-specific evaluation metrics like, say, the root mean square error, RMSE, for a numerical prediction into a smooth, continuous reward signal between 0 and 1.
I see. So a nearly right answer gets a high score, but not quite 1, while a way off answer gets close to 0.
Precisely. This continuous signal provides a much smoother gradient for the learning process, making the training more stable and ultimately leading to better performance, especially for those tricky scientific metrics.
That methodological rigor, the adaptive reasoning, the clever RL rewards, it seems that's what enables the sheer breadth of this model. Let's talk about that scope now. You mentioned 5 capability families.
Yes. 5 main areas. The first, sort of the foundation, is scientific translation. This is all about making different scientific representations talk to each other.
Like
Converting accurately between, say, an IUPAC chemical name, which is human-readable but complex, and a SMILE string, which is machine-readable. Or going the other way, generating a text description from a given molecular structure, basic interoperability.
the essential groundwork. Got it. What's next?
Capability 2 is text and knowledge extraction. This focuses on understanding language specifically, pulling structured information out of unstructured scientific text, like research papers.
So turning a dense paper into usable data points.
Things like identifying all the chemical entities mentioned, chemical entity recognition, or extracting confirmed links between chemicals and diseases, chemical disease interaction extraction.
Very useful for researchers. What about predictive power?
That's split into 2 families. Capability 3 is continuous property prediction. This is where the model estimates numerical values.
Like predicting a specific number.
Yeah. For example, ESOL prediction, estimating how soluble a chemical compound is in water. Or IIPO prediction. It's about how a drug partitions between fat and water, crucial for pharmacology. It also covers material properties, like predicting a material's band gap from its chemical formula.
Okay, predicting how much. And the next prediction family.
Capability 4 is property classification. This is about assigning discrete categories or labels, more like yes, no, or high, medium, low type predictions.
Can you give an example?
Sure. BBBP prediction. That's predicting whether a molecule can cross the blood-brain barrier, a key factor in drug development. Or classifying if a protein is likely to be soluble. Or predicting if a proposed crystal structure is likely to be stable.
Classification. Okay. And the 5th capability. That sounds like it gets into actually creating things.
It does. Capability 5 is sequence generation and design. This is where the model uses its knowledge to generate new scientific entities. Molecules, proteins, RNA sequences, materials often based on specific constraints or desired functions.
So it can suggest new drug candidates or protein designs.
Exactly. Tasks like predicting the outcome of a chemical reaction, straightforward synthesis prediction, or designing a protein sequence expected to have a particular biological function fall into this category. It's controllable generation.
That is an incredible range for a single system, from basic translation to creative design. Now, the big question. Does this unified approach with the adaptive reasoning and the reward softening actually perform better than models built specifically for just one of those tasks? The performance claims are pretty bold.
They are, but the evidence presented seems to back them up. The paper reports that SciReasoner achieves state-of-the-art performance on 54 different tasks.
State-of-the-art on 54 tasks.
Yes, and it ranks in the top 2 performers on 101 tasks overall. That suggests it's not just a jack of all trades, it's mastering many of them across different scientific domains.
Let's look at a couple of specific examples where the improvement was really significant. What about that knowledge extraction capability?
Right, that chemical disease interaction extraction task we mentioned earlier. The SciReasoner 8B model, that's the 8 billion parameter version, achieved an F1 score of 0.97.
Which sounds very high, close to perfect. How does that compare?
Well, the sources compare it to specialist large language models, which apparently scored around 0.40 on the same task.
From 0.40 to 0.97. That's a massive leap.
It is. It suggests much stronger abilities in understanding the text, recognizing the entities, and linking them correctly, pulling out those factual relationships accurately from complex biomedical literature.
Okay, a huge gain there. What about in biology, maybe with those long sequences you talked about?
Yeah, another strong area was in multi-omics tasks, specifically a task called alternative polyadenylation, or APA isoform prediction. This involves understanding long genetic sequences to predict regulatory outcomes.
Sounds complicated.
It involves complex sequence-to-property mapping. Here, the SciReasoner 8B model achieved an R-squared score that's a measure of predictive accuracy of up to 87.92%.
And the comparison.
Specialist models were reported around 59.01% for that task. Again, a really substantial improvement, highlighting its strength in handling long-context biological sequence data.
And you mentioned earlier that this strong performance really stems from that initial huge, carefully constructed pre-training data set. They did some tests to prove that, right?
Yes, the ablation studies where they test the model with and without certain components really emphasize the importance of that multi-representation pre-training.
What did they find?
For one specific task, ESOL regression that's predicting chemical solubility, a small molecule property, they look at the prediction error, the RMSE. Without the multi-representation pre-training, the error was quite high, around 94.54.
Okay.
But with that pre-training strategy included, the error plummeted by 98.7%, down to just 1.21.
Wow, a nearly 99% reduction in error just from including that mixed data.
Exactly. It strongly validates the core idea that pre-aligning natural language with diverse scientific codes like SWILES and sequences was fundamental to the model's overall success, even for predicting properties of simple molecules.
So to kind of wrap up our deep dive here, SciReasoner seems to be a significant step towards a unified foundation model for science. It successfully integrates natural language with all these different complex scientific data types, chemistry, biology, genomics, material science.
All within a single reasoning framework that aims to be verifiable. It moves beyond having separate, narrow tools for each little task.
Creating a more comprehensive system that can handle complex, cross-disciplinary problems and, based on the numbers, achieving top-tier results while doing it.
Right. That seems to be the main takeaway.
So as you, the listener, think about this advancement, here's something to consider. Science often demands solutions you can verify, steps you can check. This model's thinking mode, where it explicitly generates its reasoning steps, that long-form thinking content for tasks like synthesis or materials prediction, offers a level of transparency. How does that built-in transparency, that forced showing of its work, change how much you might trust a result from an AI tool like this, compared to just getting a quick black box prediction with no explanation, something to mull over?